So my name is Alice Defarge. I'm the Interim Associate Dean of Research and Graduate Studies, and I'm very happy to welcome you today for the Schofield lecture. So first, we need to acknowledge that the University of Guelph resides on the ancestral lands of the Atawadawan people and the treaty lands and territory of the Mississauga of the Credit. We recognize the significance of Dish with One Spoon covenant to this land and offer our respect to our Anishinaabe, Haudenosaunee, and Métis neighbors as we strive to strengthen our relationship with them. Today, this gathering place is home to many First Nation, Métis, and Inuit people, and acknowledging them reminds us of our important connection to this land where we learn and work. So, I just wanted to give you a reminder of why we're doing the Schofield lecture. It's a pretty important event for this university and for the OVC. It was established in 1970, and it brings the world's top researcher in veterinary medicine to the OVC to present their work to students, faculty, staff, alumni, and the broader University of Guelph community. This lecture series commemorates Dr. Frank Schofield, a prominent veterinary pathologist world-renowned for his work on animal disease. Schofield was a faculty member at the OVC from 1922 to 1955. Held annually since 1970, the Schofield Lecture Series has hosted many prestigious researchers who have shared their research with us, with the staff, alumni, students, and faculty members. These seminal lectures are integral in motivating OVC students, faculty, and staff to achieve the highest possible goals in research, clinical sciences, or environmental issues, and in inspiring students and faculty to become engaged in alternate career paths and become leaders in their profession. So with no further ado, I wanted to introduce Hélène Carabin, who is our guest today. So Hélène Carabin comes from the University of Montreal, who holds a very special place in my heart because I did some of my training at the CHUV, where you work. Dr. Carabin is a full professor in the Department of Pathobiology and Microbiology at the Faculty of Veterinary Medicine and in the Department of Social and Preventative Medicine at the School of Public Health at the University of Montreal in Canada. She also holds the prestigious Tier 1 Canada Research Chair in Epidemiology and One Health. Her research program focuses on the implementation of large-scale studies and intervention to better understand risk factors and burden of infectious diseases to evaluate control programs with an emphasis on zoonosis and diseases affecting the most vulnerable populations. Our research projects typically adopt a One Health approach and incorporate aspects of health and social sciences. So she plays a key role in several Canadian networks and associations that promote the use of One Health to improve the governance of infectious diseases and antimicrobial resistance, Global 1HN, in modeling emerging infection or understanding emerging zoonosis. She leads the research group on the epidemiology of zoonosis and public health, co-leads a network of the prevention of crises affecting the health of people and animals, and is a commissioner on the One Health Lancet Commission. She's the fellow of Canadian Academy of Health Sciences and an external member of the National Academy of Medicine. She has published more than 160 peer-reviewed research articles and 10 book chapters. I'm lucky I have some notes. Welcome, Dr. Carabin. Thank you so much for your presence here. We're very honored to have you with us. And with no further ado, I will give you the floor to present your conference.
Thank you so much. Okay, hi. So today I wanted to talk to you a little bit about what I've been doing in the past 6 years after I came back to Canada after spending 16 years in the US. And what I've been doing is trying to understand a little bit more what the role of epidemiology could be in the bigger One Health picture. So first of all, let's start with a few definitions. I'm going to talk about One Health and epidemiology. So we need to know what we're talking about. One Health, you probably all know what it is here, right? The whole lab has finally come up with a definition after I've been in meetings over and over where we would argue over 2 or 3 days what the hell is One Health. Now we know, all right? So One Health is an integrated, unifying approach that aims to sustainably balance and optimize the health of people, animals, and ecosystem. And plants are within that as well. And what I really like with the definition is the visual that comes with it, where it shows that people typically work in sectors. You know, it could be environmental health, human health, animal health, and then within that they subdivide into more silos and silos and silos and so on. Okay. And then on the other side, people and animals and plants live in society. And they're organized at all sorts of structures, from really local all the way to international structures. And this, well, usually is pretty much decided by humans, like it or not. And the idea of One Health is to put all of this together in a big mush and get people to talk and then try and coordinate things, communicate, collaborate, and train, obviously, do capacity building, so that we approach problems differently. And we approach problem by recognizing that we cannot only maximize the health of humans. Even for human health, that's not good. That we need to really think about maximizing the health of everything in order to improve, bring everything up. That's my vision of One Health, and that's where also, so I didn't do the language acknowledgement, but this is where I see it. You know, indigenous people, be them in Canada or where I've worked a lot in Africa, they see this. You don't have to explain One Health to them. They live it every day. And they're going to tell you, when there's too much water, it's not going well. When it's too hot, it's not going well. When I lose my cattle, my kid's going to die. And I'm going to vaccinate my cattle before I vaccinate my kid. So we need to recognize this understanding of things. And this is where I really view my land acknowledgement and acknowledgement of knowledge of traditional people. So epidemiology, I had fun when I did this because I realized that there are 102 definitions of epidemiology in the literature. So we have one in One Health and we have 102, of which only 9 in veterinary medicine. So we're doing a little bit better. We don't argue as much. But, you know, so I picked 2. The Merck Manual of Veterinary Medicine says it's the study of diseases in population and factors that determine its occurrence over time. Yeah, fair enough. And the WHO, the study of the distribution and determinants of health-related states or events, including disease, and the application of this study to the control of diseases and other health problems. It's pretty complete. And what I really like with the WHO, they don't say human population. They just say populations. And that's what we want to do, is understand the distribution of factors that may affect health. Now in epidemiology, often what we try to do is to determine the extent to which a certain exposure has a causal relationship with an outcome. In order to get there, it's very complicated because there's all sorts of things that can go wrong. So first of all, just if you don't study enough subjects, there can be error that occur at random, so you're going to conclude something that's not right because you just didn't study enough people or animals or whatever. The second thing that can go wrong, so let's say you have a large sample size. Well, when you study your population, you can make mistakes in classifying them as being exposed or diseased, misclassification error, or there can be other factors that kind of is going to make that your association between the exposure and the outcome is not correct because that other factor is linked to both the exposure and the outcome. That's confounding. And then the third thing that could go wrong is you may not select the people or the animals in the right way. You might select, for example, more animals that are exposed than diseased in your study. That's going to increase the level of association between your exposure and your outcome. That's selection bias. So if you do all of this right and you have no bias anywhere, then you can do causal inference. So that's very nice. However, we're trying to do this in the frame of a zoonosis, all right, where the stages of the disease can be in pigs, it can be in humans, it can be in the environment, and there's a cycle. So all these biases I just talked about, and the association between an exposure and an outcome, you have several outcomes in different species, and the same exposure may be affecting all these outcomes, but the levels of bias are not going to be the same in the whole system. So you can imagine when you try to link epidemiology and One Health, you have to try and think about all these aspects, and it's really not that obvious. So when we try to apply it, what we try to do is to try and understand these complex associations between determinants and health statuses in the different species and sometimes the environment and plants, sometimes. We have to recognize how well our tests are measuring the outcome. So the same infection may not be measured as well with exactly the same test in pigs and humans. So you have to take that into consideration. The way you sample the population in order to do causal inference may not be the same for humans and animals and plants, because the epidemiological unit may not be the same. You may not try to get to the same information. So for example, if you want to link infection of schistosomiasis in dogs and humans later on, a dog is not linked to one person. A dog is linked to a population of people. Therefore, you have to sample accordingly to be able to make that link. And of course, you have to think about the behavior of people and animals. So thinking about cysticercosis, which I've worked on a lot, male pigs will not behave the same way as females that have piglets, and they will not roam as far. Therefore, they may not be as exposed to the infection. So behavioral in both animals and humans are important. All of this has to be done by engaging with stakeholders, with communities, which brings all the social sciences, anthropology parts. And then we have to think about all of this is organized in the system. Easy, huh? All right. And on top of this, and I'm not expecting you to read any of this, but the point is we did a review of the PhD programs that were offered around the world. And as we were doing this review, we noted that one thing that was really not covered enough is definitely the social aspect of One Health. And also the fact that we have to rethink of health in general in terms of socio-ecological determinants of One Health. So in human medicine, or WHO speaks of the social determinants of health. You need to add the ecological part in One Health. So I'm going to talk about 2 case studies, maybe 3 if I have time. We'll see how we're doing on time. It's there, we'll see. So first of all, let's go for it. So I talked to you about cysticercosis. So briefly, humans are the definitive host. They have one very big segmented worm in their intestine. It could be 2 meters, something like that. People are going to shed the eggs in the environment with open defecation. The eggs get picked up by pigs. Pigs eat human poop and they make bacon with it. So it's fantastic, very ecological. However, the problem is they get infected with cysticercosis. Human eat undercooked meat, they get the big worm. For human health, no big deal. Even for pig health, no big deal. The problem comes when humans get infected by human feces through food or bad hand washing, whatever, and then they get infected like the pigs with the larval stage, cysticercosis, and this cysticercosis tends to go to the brain. So that's a brain, every little hole in there is a larvae, a cyst. And it tends to be associated with epilepsy and a whole range of neurological disorders. And it's everywhere in the world where there's poor sanitation, poor vet services, and pigs, basically. You could say that. So, however, if we want to try and prevent cysticercosis, it causes epilepsy, which is hugely stigmatized in underdeveloped countries. There's no reason why people should have epilepsy caused by that. So let's try and find factors that are causal, that cause infection in humans or pigs, so that we act on them, we cut the cycle, and we better the health of both pigs and humans. However, normally in order to identify, to control for confounding, so these other factors that can kind of confound the association between the exposure and the outcome, you do a DAGS. A DAGS is a directed acyclic graph, right? So normally you figure out, okay, is there a cause between the exposure and the outcome? There might be a confounding factor that makes that association not correct if you don't adjust for the confounder, or sometimes you have something called a collider. They're not confounder. If you adjust for it too much, you're going to have an error in your association between the exposure and the outcome. Except that here, we're not acyclic. There is a cycle in cysticercosis. So if we try to figure out the association between an exposure and an outcome, and there's a big cycle going around different hosts, how can you do that? We started by doing this. Oh, yes, we'll do a DAGS for cysticercosis. Oh, wait a second, we can't. So I have a student, Elias Jackson, that's started working on this. So first of all, the first part was to figure out how can you do a DAGS for infectious diseases, particularly zoonoses. And I'm not going to go through a long explanation, but the goal is to split the time so that you only go through one cycle of the life cycle, and by doing so you can identify the factors that happen at these different stages during one cycle. So if you want more details, it's in the paper. And then we said, okay, nice. Most people who do DAGS, they just do DAGS. But we said, well, maybe it should be based on the literature, right? We cannot just assume what factors are associated with what. So we, primarily Elias, did a massive literature search on all associations that are involved in the life cycle of cysticercosis. So risk factors for pig cysticercosis, risk factor for human cysticercosis, risk factors for human tetaniasis, risk factors for eggs in the environment, noting that there's hardly anything in there. So he found 159 studies that were included, and this is just showing the distribution mostly in Africa. And then we rated the quality of each association, and there were 883 associations that were rated. Most of them were weak, so the evidence wasn't very strong. And then the other half was strong or moderate. So with this, created a DAGS. This is only showing the pieces of evidence that are strong or moderate. There's another DAG that's crazy big that has all the potential links. But by having this, then we can determine, so, this one was for human cysticercosis. And then you can choose, okay, which exposure am I interested in order, as a potential factor I could act upon in order to prevent infection. And then you can identify which one. So for example, it could be latrine use. And then identify which factor you have to adjust for in order to measure the association between latrine use and human cysticercosis without bias. And so what? Okay, so why go into the trouble of working on this for 4 years to come up with that messy thing? Well, it's very useful because then you can identify a minimum set of confounders that you have to control for when you do your analysis, so you get closer to the truth. And second, now the third thing that Elias is doing is taking this DAG and using it to develop a causal Bayesian network. Causal Bayesian network are helpful when you try to identify the effect of an exposure that may occur several steps before the outcome of interest, and then determine if I change it by that much, what is going to be the impact on my outcome of interest. And you can put cost to that as well, in order to say, okay, how cost effective would that be? How cost beneficial would it be? So that's where we're at right now. And the causal Bayesian network for pig cysticercosis is being finalized as we speak. It's been long, but we're getting there. So the second example I wanted to talk about was what about the One Health surveillance? One Health surveillance in 2024 with social media, modern tools, and how helpful that might be. And that was a huge program that, of course, occurred because of COVID. You know, we had to do other things during COVID. As an infectious disease epidemiologist, like, okay, I hear the call. I worked a lot with FAC, with Public Health Agency of Canada, to try and find solutions. So the context is that there's more and more evidence that Twitter can be used, well, was usable before, now that it's sexist something else, but there'll be something else, maybe blue sky, who knows, there'll be something else. The extent to which social media can be used to identify emergence or events that are threats to public health or animal health. So that was used, it has been used for swine flu, as well as avian influenza, and I think that was a group, that was a group actually here, right, in Guelph. And we also all know that the first case of COVID was shared through social media in China. But it did get picked up, but a bit too ignored. Maybe we shouldn't ignore it as much. So, in January 2020, Erin Rees, my colleague from FAC, decided, you know what? Maybe we should start downloading tweets. So she started with a student, downloaded tweets, and ended up downloading 281,487,148 tweets between January 21, 2020 and August 23, 2021. The idea behind this being that there's a thing in Canada called the Global Public Health Intelligence Network that I will call GFIN. The idea is to identify events that are of concern to public health. And right now what they're doing is they're mostly getting data from official media, some tweets, but only from officials, so from ministry, governments, and all of that. It gets filtered to a certain extent through some AI. It's semi-automated. And then there's some analysts that go through manually through the different not alerts, but the articles that look suspicious. They review them, and then they will report if there's an alert of something that should be looked at more in depth. So one of our first question was, can we, whoa, I don't know what's going on. Can we add public social media to this system? Is it feasible? Is it useful? There's a lot of infodemic. There's a lot of crap in social media. So would it be helpful to do this, and could we detect an event faster by doing this, like what happened in China? So with COVID, indeed, that's what happened. It was that Sina Weibo post that happened in November 2019. If we had picked up before, maybe we could have acted earlier. The second question is, well, should GFIN start integrating animal health surveillance into their system, since 75% of emerging diseases are zoonotic? Well, it would make sense, right? But in order to do this, you have to make sure that the people in charge of animal surveillance agree and understand the consequences of this and understand how they feel about it. So that's how we came up with a big list of objectives. That's like 5-6 years of worth of, well, since COVID. So first of all, we wanted to know understand the map of information flow between human and animal health during one health crisis that was stopped. One case of swine influenza in I think it was October 2020. Then evaluate barriers and facilitator to incorporating animal health and personal social media in something like GFIN. It doesn't have to be only GFIN, but any event-based surveillance system. And then after that, we continue to dig into it to try and figure out, can personal social media, well, first of all, does it reflect what people do in Canada? If you use Twitter, does it mean anything? If people are angry, are they really angry? Does it reflect something useful for public health? Or it could be for animal health, as a matter of fact. And then also figure out, okay, what if we were to use personal social media, add personal social media and information from Google or something to GFIN, would we detect emergence faster? So is it worth it? So all of this was funded by all sorts of funding agency because it took a long time. We worked on it. So the first thing we did was to try and map the communication channels during a case of swine influenza, as I said. So we did that looking at documents, mostly from CAS, the Western Swine Health Influenza Network. Did some semi-structured interviews and then analyzed it with thematic analysis. So I'm not going to go through this, don't worry. That's the flow of information during that case. But what came out of this primarily is that we had governmental stakeholders that were primarily led by human and public health, but they collaborated with animal health. And then on the other side, you had the industry animal health stakeholders. And then in the middle of this, in order for this case to be acted upon really quickly, they had for them that the data was available very quickly. They had a pre-existing relationship. They trusted one another. So the industry trusted public health trusted industry, primarily because they had a public health veterinarian. And that really, really helped in this case. And we should do that in every province, honestly, because it really is a big plus. And plus, it was a notifiable disease. Influenza is well known. It's not like COVID, we didn't know at the beginning. But all of these are really important factors. And one quote I just wanted to put on there from a person that was interviewed about sharing data from animal surveillance to human health surveillance, is that your perspective here is spread or transmission from swine to humans. But I think that what happens more frequently actually is spread from humans to swine. I don't see that we could get anywhere closer before we also start to think about the danger people actually have to the health of swine. All the time you have transmission the other way from human to pigs, and that's sort of ignored. So the bottom line is, if we want to do this, it has to go both ways. It cannot be only for human health, and that's what I said at the beginning. That's not one health. That's not going to work. It has to be both sides. And what's interesting is after that quote, after all that work, there's a paper that came out, I think it was in March this year, that demonstrates that there is more viruses that jump from human to animals than the other way around. So we've got to change the way we think, right? Continuing on, now we did our case study on COVID, which was a complete mess. But same thing, documents review, semi-structured interviews, 9 deliberative dialogues, and we ended up with 770 pages of transcript to try and make sense of it. And I'm not going to go through the figures because it just, I would spend all day on this. But it's just an idea of how it worked. There's some animal health actors here with CZ. There's some WA international level, public health, etc. But I'll stop there. So, in terms of barriers and opportunities for incorporating animal health and social media data into GFIN, for the animal data, people recognized that it was important. That's a person from FACT who told us that for potential public health threat, the analyst will review those to make determination about whether or not they should be in the system. And the analyst will look for a wide range of potential public health threat that include infectious disease, radiological or nuclear as well. And it does cover both animal and human health. However, one big barrier is that there was a lack of communication and a lack of training of the analyst to see which animal infection could be a threat for human health. And in terms of personal social media, while they recognize it was faster, easier maybe, but the quality of information is a problem. It's hard to evaluate. There's too much information. It's not manageable. And it would need automation and training. So I'm not going to go through the quote because of the time, but so what we recommended was more training for social media, explore strategy to extract data from several type of platforms, especially now with Twitter, which is X, which is not all that helpful. Maybe we need to learn how to dig more into Facebook, but then there's all ethical issues that you cannot just go into the page of anyone. Even Twitter, you know, when people start using Twitter, they don't really know that it could be used for all sorts of things. So there's a whole big bubble of issues with this, but it can be useful. And then that when you use algorithms to extract information, we have to make sure the context, the socio-cultural context is taken into account. So all the work we've done was in English. So we're missing all Francophone Canada. And for those that were in French, they were automatically translated. Well, forget about sarcasm, forget about expressions, it's not going to work. So, next question, who uses personal social media in Canada? So we ended up doing 27 cycles of surveys with Angus Reid between September 2020 to March 2022, except during the elections and other. It's a forum of Angus Reid. They answered questions about social media use, Twitter use, and what they tweeted. But also, we asked them questions about how they behaved. So in the past 2 weeks, did you use a mask in public? Were you vaccinated? Or recently, or before vaccination, do you agree with the vaccine? And there was also social distancing, but that was super hard to measure. So we ended up doing mostly mask and vaccination. So overall, there were about 21% of respondents using Twitter. These are weighted for the Canadian population. A lot less in Quebec, more in Ontario, less in females, less among people of European descent. Which was a bit of a surprise to me. And there were a whole lot of other differences of who uses Twitter or not. So the Twitter users who tweeted about COVID are not representative at all of Canadians. So we have to be aware of this when using Twitter. I'm continuing my slide that you're not seeing the nice pictures, but basically, Canadians were pretty good during COVID, right? About 90% were wearing masks. And more than 90% ended up being vaccinated. So it's pretty good. when I was in the US in Oklahoma, it wasn't there at all, right? And then the other thing, it's there, good. The other thing is people who tweeted about COVID, they had poorer compliance to vaccination than those who tweeted about something else and non-Twitter at all. So those who tweeted about something else than COVID had better adherence to vaccine than those who didn't tweet, and those who tweeted about COVID had worse. Because when they tweeted, they were usually not very happy. So the next question is, do they walk the talk? In other words, if they do tweet negatively about public health measures like masking and vaccination, do they actually wear masks less and get less vaccination? The answer is yes, and very much so. So that's, however, interesting, because it's the first time that a study demonstrated at the individual level that that was the case. That if you say, I hate vaccines, well, you're not going to get vaccinated. Oh, stupid people wear masks, you're not going to put a mask on. But that wasn't the same amongst Anglophones and Francophones. So when a Francophone is angry, a Francophone is really angry. Well, we know that, right? Like, I'm not going to wear the mask. So the link between being angry on Twitter and not wearing the mask was much stronger among Francophones than Anglophones. For vaccines, that difference wasn't there. However, those who were supportive about Twitter were vaccinated a whole lot more than those who didn't tweet, and those who were critical, they got vaccinated less. These are odds ratio, so it's an odds ratio of 4, so maybe 3 times roughly. Okay, so then, do sentiments shared on Twitter represent what the general population feels? No. Why? Because Twitter users are not representative of the population. However, could Twitter be useful for public health or for One Health? Well, we've got X now. So I don't know, honestly. Probably not. It's probably really not the same story. There might be something else that will come up, again, blue sky, but it would be a bit dangerous to say, oh yes, we can use X the same way as we could use Twitter. And then could sentiment share on social media be measured by AI? Right? Because you cannot go one by one every single tweet. You have to use artificial intelligence. Well, that was the next question. So, I don't know why it does that all the time. Bam. Okay, stop there. So the question came because with Aaron, we did an analysis trying to link these stringency index of public health measures. So how stringent were the public health measures in Canada, across Canada, in the different provinces, and the link with RT, so transmission of COVID. And what we found is there was a lot of variability among provinces, that it just wasn't very consistent. So then we wonder, well, maybe it's because it depends on how well people follow the public health measures. And since, well, maybe we can measure it with looking at tweets through artificial intelligence, maybe we could try and understand better why the stringency index wasn't working the same way everywhere. So that was the next question. So what we did is we compared tweets about talking about different topics related to public health measures, and then tweets with sentiments regarding public health measures. And we tried an AI algorithm that was developed, I think it's 2019, to measure sentiment. It's a whole AI thing. I didn't understand all of it, but it does valence, complicated word. And we use Bayesian class models, hierarchical Bayesian class model, not assuming there's any gold standard, comparing this to MTurks. MTurks are people reading tweets and evaluating humans, what they talk about and what the sentiment is. So we did that. We submitted the paper, got rejected. And they said, why don't you use GPT instead of the, the artificial intelligence algorithm? And they were right. So we redid the whole thing with ChatGPT. And what we found is both the algorithm and ChatGPT were pretty good at classifying topics. So the sensitivity about topics about vaccination with ChatGPT was 91%, specificity is the same. The ChatGPT was always doing a little bit better than the algorithm for topics, but the topic was not too bad. And then in terms of sentiment, so that's where that valence thing I was talking about comes into account. You can put different threshold of valence to determine if the sentiment is positive or negative. And depending on the threshold, you decide, okay, this is a positive sentiment, this is a negative sentiment. So what this figure shows basically is that ChatGPT was as sensitive as our mTurks. So the different letters are our mTurks to classify sentiment, but it was a little bit less specific. So they were more false positive than the mTurk. On the other hand, the algorithm was terrible. It's really terrible. It could not classify sentiment. So next step, we'll use ChatGPT to try and understand what we were trying to do first with the stringency index. How am I doing? I'm not too bad. Okay. This one was then to see, okay, what if we add social media information and Google Trends information into a model to predict when the emergence of COVID will occur in a health region of Canada, 10 days before what GFIN would have reported. And it's a super complicated model. Do not ask me questions about it. I don't understand it. It's called Deep Gated Recurrent Unit. There's a geek in computing that did that. But basically what we showed is that each time we add information from Twitter or Google searches about COVID, the sensitivity to identify the next region where COVID will emerge 10 days before improved. So yes, social media information can be useful to predict emergence, knowing, however, that we had an idea at the time what the symptoms of COVID was. If it were to be a really, really, really emerging disease that we don't know what the symptoms are, however, could we find something that animals have diarrhea and humans have diarrhea, and then, I don't know, something's going on in the environment, and then you put all of this together? Maybe. I don't know, but that's for the next question. So, again, next steps, as I said, is to review what Erin had done with the stringency index. I would like to see the extent to which social media could contribute to the surveillance of events in a One Health context. But then we would have to go through something else than Twitter. But I'm sure, vets, I don't know if you have that in Ontario. In Quebec, we have Vétérinaire du Québec. It's on Facebook. It's not just vet that put different problem clinical cases, but other things too. At one point, there's a lady that put a picture of a puppy that had vomited worms, but then she was smart enough to send it to the lab, and it was Billus ascaris in the middle of Montreal, in a family with kids. And then if you end up with a lot of kids that have epilepsy or neurological diseases, I'd be very curious, because no one cares about Billus ascaris. And then what about companion animals? There is no surveillance for anything except rabies in companion animal. Yet, who sleeps with a cat or the dog on their face? Right? So you need to do something about this, because it could be the next pandemic. They've shown that AMR in tartar of teeth of dogs and humans were linked. Right? They share bacteria that are resistant to antimicrobial on their teeth because they sleep them, you know. Okay. Do I have time? Can you give me a few minutes extra? Yeah? Okay. All right. So this is a bit on the side. That's the other project that I was keeping for the end. Remember during COVID, we said dogs could sniff COVID. That'd be really cool. Nothing was going on in Canada. So at first, répondre à vos questions, that's a research group I'm involved in as well, one of the too many things I'm doing. And we, the idea was to inform public health workers and others about different questions. So we did a review, to see if dogs could be helpful. And what we found is there was a lot of variability in the training of the dogs and a lot of variability of how they were measured. So we decided to start doing something about it and got funded, of all places, from the Ontario Ministry of Education and Universities. It was impossible to get it in Quebec. It was impossible to get it at the federal level. But Ontario said, yeah, let's do it. That was great. So we worked with UHT and University of Toronto and managed to get 3 sweat samples from 2,354 people that were being screened for SARS-CoV-2 between October and December 2021. Of those, 121 were PCR positive. The others were either negative, and another 13 went from negative to positive because they were tested twice. We ended up having 3 dogs from one handler, and then the dogs had to go through different steps that I'm going to show in a minute. And I'm going to show the analysis of validating the dog to know what's the sensitivity and the specificity of the dogs to detect COVID. You've probably understood by now I'm interested in bias, right? Okay. So this is a little video of the different steps. The information is in French, so I'm sorry for those who don't understand. I'm sure you can figure it out. So the first thing to learn is that when you go in front of what you're supposed to sniff, you get a click and you get a reward. But you know, the dog has to learn that. Second was to learn to sit on a mat when you're done. Third thing was to learn to recognize COVID. Remember, we only had 120 positives. That's difficult. Same thing, but in another room. We had to change room anyway. I won't go through. The dogs were getting fat, so we started giving them bottles. The same.
Yes, sir, good girl, good girl. What is that?
And then if the dog finds one, he has to go again. So he has to learn to do this because there'll be several samples on the line. So all of this takes a lot of time. So then that same thing, but now we had the station. It took a long time to get the sniffing station. It's complicated. Oh, and then they had to learn that it's okay to be all negative. You know, the prevalence is maybe 10%. That's jazz, by the way. And then that was trying to detect it.
Yes, sir. Yes, sir. Good girl. Good girl. Yes, sir. Okay, that's it. Yes, sir. Good girl. Good girl. Yes, sir.
Now the handler didn't know which one were positive, and then it started not going so. And he shouldn't be next to the dog. He should be letting the dog do his job. But and you can see he's frustrated. Okay, so that took 5 months, I think. And we had to stop in advance because our handler just couldn't take not knowing how well the dogs were doing. Anyhow, we still had data. But we ended up doing a very complicated Bayesian latent class hierarchical analysis where we took into consideration the fact that the PCR and the observation of the behavior, come back to the behavior of the dog, is not perfect. And that the fact that some sample had to be reused was a factor, but that's in the literature. It happens all the time, but they don't tell you. And that the fact that the videos that were observed to judge if a dog was marking or not were viewed several times. And then on top, the observation was done with or without sound. There's a dog effect, obviously. And there's a video assessor. We had 2 people assessing videos. And most of the literature, they don't even give you all these details, so you can't tell. So what's really interesting is, so I'm taking the example of one dog, Mathieu, one of the observer, looking at the dog without sound. If we do a normal analysis, frequentist, you assume PCR is perfect. The sensitivity of JAS is about 81%, specificity 78%, which is really not bad. It's within the range of what's been reported. If you rerun the analysis, you assume the PCR is imperfect. Actually, it gets even better. You're at 85% of both. And that's, again, what you find in the literature. But when you take all of this stuff into consideration, the dog is not so good anymore. It drops to 52%, and the specificity drops as well. So the idea that we had that the dogs were really, really good during COVID, well, maybe not that much. I'm the first one who's not happy about this, but it is what it is. And maybe it's because we don't train them well. There's no standard. It's not done rigorously, and it's not reported rigorously. But dog, they could be an amazing tool. We're talking more and more about lack of access of screening for breast cancer in remote communities. Imagine if you could have dogs. Oh, yes, she should go. She should travel for a mammography. The others are fine. That would be fantastic. But I don't think we're there yet. So, to conclude, complexity can lead to really important biases, and we have to learn how to adjust for these biases in the context of One Health. Misclassification error is often neglected, and there's even papers saying this. It's time we start really looking at this with the appropriate tools and the appropriate epidemiological design within a One Health vision. It's absolutely essential to engage with stakeholders. What we learned talking with people with animal surveillance, if you don't do this, you will not have a One Health surveillance. And we have to listen and adjust accordingly. But all of this takes a lot of steps, a lot of time, and a lot of money. Doing One Health is super expensive. So what I want to do for the next 7 years in the next CRC, which I submitted in October, I'm waiting, in Canada is to start looking at neglected parasitic zoonoses, the lack of surveillance for companion animal, Lauren and others, and impact of those zoonoses on mental health. which also is something super complicated, super neglected, of marginalized population, and this is what drives me. It's very complicated because the diagnosis of bad. So another thing we want to start looking at is the lack of surveillance for zoonotic diseases in companion animals and national security. So we put a CSSP proposal with Erin, still my partner in crime, to try and look into this. Most tests are done by IDEX, and the data is owned by the Americans. Trump just got elected. If they detect something really bad in a dog or a cat or something, where is the information going to go first? Are they going to close the border before we know it? I don't know. So, and then the third is we just got funded to start a One Health cohort in Quebec through CIHR One Health grants. So we will go into 30 dairy farms and 20 backyard farms, take samples of milk, cats, and other animal in the backyard farms, and test for exposure to avian influenza. Do the same with humans, and take environmental samples with the idea that add other types of farms in the future and go back. And create these cohorts that are really One Health, with which we could detect anything. Contaminants, other infectious agents, but together, right? Not, we study human here, we study cattle there, we study environment there. Put them all together to have way more power to detect what's going on. And then in low and middle income country, I'm still working on cysticercosis. That's never going to end. And we're starting to look at misclassification error in estimating, of imaging actually, because neurocysticercosis, when it's in the brain, the gold standard is image, MRI or CT, and everybody assumes it's perfect, but it's so not perfect. There's so many problems with this. So I'm starting to tackle this, because if you don't tackle this, you won't know what the real burden is. And until we don't know what the real burden is, it will continue to be neglected. And people will continue to suffer from this. So anyway, Illuso, Marine is here with Jane, Heather, maybe somewhere. So that's a project in Senegal and Burkina Faso where we're trying to work with women and girls on zoonoses, trying to know what their priorities are and trying to help them to start controlling a little bit. We won't change everything. It's a pilot study, but it's huge. We cannot talk about it officially because it hasn't been announced. And then the final one is looking at the impact of climate change on parasitic zoonoses and small-scale farming by women and girls. I don't know if you've seen that FAO report on the unjust climate, but it's very clear that climate change is affecting farming by women a whole lot more than what men do. And it's time to tackle that and see if we can help the women. That's it.
Thank you very much, Hélène. That was so passionate. I wish I had you as a professor in vet school. I may have changed my path, actually. So it was really, I hope it inspired a lot of people, I'm sure, in the room. We'll take some questions first, and then we'll move on to a session where you will be able to meet like Hélène personally. So yeah, Travis, I'm sorry. I'll have to repeat your question for the online people, but we have only one microphone, so.
What was the reaction to the Public Health of Canada, the recognition that maybe animals are more at risk from human than the other way around?
So the question is, what was the reaction of the public health folks to the comment of the animal health people stating that influenza probably goes from human to pigs more often than the other way around? Well, very interestingly, they didn't answer to our invitation. So we do not have the point of view of public health. I'm not, we know that. but we need to try and do something about it. So why didn't they participate? This was a finding of its own. So I don't know, but I hope they will realize it more.
John, you had a question?
Good talk. Thank you very much. I wonder if when you dug into the data on the separation between people who are highly vaccine hesitant and people who bought on the body early and were happy to get vaccinated and that. So basically, following public health instruction versus not following public health. Were there any other pieces of data that suggested, like any other things that came out in your mining of that data that would suggest cause or point to rate the common theme that might explain why that happened? And that's our failure as One Health scientists. Yeah. That there's been such a big emergence in the last 10 years of people who are resistant to what appears on evidence to be good advice.
Yeah, so the question is if we looked into the data, other variables that could have been linked to poor adherence to mask wearing or vaccination. The answer is actually no, because a lot of other people are looking into vaccine hesitancy and these types of questions, and we really wanted to look at Twitter. However, we did adjust for potential confounders that we could find. But no, that's not something we looked into.
And nothing kind of popped out. I mean, I know it's unscientific, but sometimes themes emerge that, that you have to investigate with data-driven science, but were there trends in regional trends?
Oh, yeah. There were tons of variation between provinces, between Francophone, Anglophones, aged, gender. It really wasn't uniform across Canada. Well, the West tended to be a bit worse. Quebec tended to be a little bit better. Atlantic provinces, I think, were top, if I recall.
Any other questions? We have time.
So I'm just wondering, how do you incorporate a One Health approach when you're investigating, like basically lab sciences? I understand that a lot of work in you're doing a population with so many different factors. Well, for example, my work in chronic pain, I isolated lab mice, and their whole world is this little cage. For people that work in cell cultures and you're trying to draw conclusions from that, like have that impact on the population. How do you go here that sometimes incorporate One Health perspective?
Ooh, that's a difficult question. So the question is, how would you incorporate One Health in more the basic science work, such as working with mice, reaction to pain in the lab? Well, it's all the hierarchy, right, of knowledge. I guess, and I'm really not certain of my answer here, but I guess, obviously if you find something, we're all mammals, right? If you find something in the mice, you may ask the question with databases in human medicine, depending what you're testing, and try and see if that link seems to be there, or talk with clinicians or researchers doing similar things in the human side, and then you get more into the comparative medicine side of things. This is part of One Health, and I think that would be the first step to go further. That basic knowledge is essential to go further, right? So for example, to understand if PFAS have some impact on cancer or whatever, often you have to try and test things on cells. And that can bring a hypothesis at larger scale later on. But there's definitely a role to play, and try again to get in contact with people working on similar things in different animal species, in humans. That I think that's what I would say. It is. It is a first step. We need to know how things work, right? If you don't, can't get anywhere.
Any other question for Hélène? Do we have an online question? No, you're not a speaker, you're just intimidating. So you will have also the opportunity to ask questions to Hélène. We'll have a Slack. So I just want to finish the question session by thanking also a lot Katie and Carrie-Anne for organizing this event. Thank you for fixing the IT, Katie, on the fly in the middle of the talk. Thank you, Anna, for being here. And then I wanted to invite Madison to present the medal to Hélène. So Madison is the president of the CDSA, and she will present you the Medal of Schofield. Thank you again for coming today. So now I invite everyone to go to the back of the room, and you can ask questions to Hélène. You can have a snack with us. Thank you so much again for coming today, and thank you again, Hélène, for coming. And it's an honor to have you here. It was very fascinating.